Good morning, folks. Welcome back to our notes for AP Bio on Unit 7. Today we're getting into part 4 of the notes where we'll talk about topics 7.9 and 7.7, which is on phylogeny and common ancestry. Let's jump in. We'll start with 7.9 phylogeny. So phylogeny is the evolutionary history of a species or group of related species. So it shows you, who evolved from whom, where there was a speciation event, common ancestors, that sort of thing. We usually depict phylogeny as a phylogenetic tree to show those evolutionary relationships. Like down here, we can see here's the ancestral lizard with all their limbs, and then there's a speciation event here, and some of them evolved into what today we call geckos. The other branch eventually evolved into all these other things here. So we can see how closely related they are evolutionarily. Taxonomy is very similar but slightly different. It's the study of classification and naming of organisms. And the two don't always match up perfectly. They usually match up pretty well. This is strictly focused, phylogenetic trees are strictly focused on evolutionary relationships. Taxonomy is focused on common characteristics, which often comes from common ancestors and phylogeny, but not always, especially since taxonomy was developed before we knew about genetics and before we really knew about evolution. There can sometimes be some disconnects in there. So let's review binomial nomenclature, which was developed in the 18th century, so the 1700s, so before Darwin. A gentleman by the name of Carolus Linnaeus published a system of taxonomy based on morphological resemblances. In other words, the physical features of the organisms. We still use two key features of his system today, the two-part names for species and hierarchical classification. And we covered all this stuff in Bio 1, so just a quick rehash on it here. So the two-part scientific name of a species is a binomial. So we call his system binomial nomenclature, meaning two name, binome, nomenclature, naming system. So a two-name naming system. The first part of any scientific name is the genus that the individual belongs to. And the second part, called the specific epithet, is unique for each species within that genus. We always capitalize the first letter, and the entire species name is usually italicized. If it's not italicized, it's underlined. So both of those groups, their names go together and make the scientific name. So if you know the genus group that somebody belongs to and the species group that it belongs to, you can figure out the name. So ours is Homo sapiens. We belong to the genus Homo, and our species within that genus is sapiens. What it literally means, if you look at the meaning, is man wise. There have been other organisms in the genus Homo on this planet. They're just not around anymore today, as far as we know. So Neanderthals, Homo neanderthalis, Homo erectus, Homo florensis, a few different ones that were very closely related to us, but are no longer here. They're extinct. The other thing we use is hierarchical classification. So that's the kingdom, phylum, class, family, order, genus, species. Order, family, genus, species. Can't say it too fast. So when Linnaeus first came up with it, it started with kingdom, phylum, class, order, family, and then genus and species. More recently, we've added in domain to account for some of the new information we have and make it a little bit more useful. So a way to remember this, the one I learned when I was in school, we didn't worry about domain back then. We just called it king's play chess on fiberglass stools. So kingdom, phylum, class, order, family, genus, species. Now with domains, we got to add in a D word at the beginning so you can make a question, do kings play? Or you can make it a statement, dope kings play chess on fiberglass stools. It's up to you, but it's a way to remember it. You may want to come up with your own. You may want to use mine. Each of these groups is called a taxon. Okay. Up here we have broader taxons. Down here we have more specific taxons. So kingdom, there's all sorts of different things in a kingdom, right? From humans, other mammals, reptiles, birds, insects, sponges, sea slugs, fish. They're all in the animal kingdom. But as you go down, we get more and more specific, like we're class Mammalia. So here we've got, we all have backbones, we have fur, we give live birth, we feed our young milk. That's what we have in common. You go even further down, you get even more commonalities, and you get into primates and those kind of things. All right. Here's just a show of how these things are kind of like Russian dolls nested within each other. So you got your species, and species belong to a slightly bigger group, the genus, with other things that are similar, like lions and tigers here for this panther, and they belong within a even larger family, okay, which would be other cat-like things, Felidae, and they belong within a larger one called Carnivora in the order. So that would include things like bears and wolves and stuff like that are all here within the larger class of Mammalia. So here they in the same group as us. Then we go up to a larger group called Chordata. So that everything in here has a backbone, or a spinal cord, I should say specifically, not a backbone necessarily. There's a subphylum called Vertebrata, which we won't get into. And then we belong to the animal kingdom. And then finally, we belong to the domain, which is based on our cell type, which is domain Eukarya. And there's also domains for the two different types of bacteria, eubacteria and archaeobacteria. For a while, it was just two domains, the eukarya and prokarya, and then the two bacteria types were split into kingdoms. I think there's still some debate on that, but our book uses this one, and that's the one that the AP College Board says we have to use, so we'll use this one. Just a little bit of a video kind of showing that split. Meet you on the other side.
In the early days of biology, all organisms were classified as either plants or animals. Single-celled eukaryotic organisms such as algae and paramecium were assigned to one of the kingdoms according to whether they are photosynthetic or not. Fungi and prokaryotes were grouped with plants because fungi are sedentary and prokaryotes have cell walls. A five-kingdom scheme was devised because some organisms don't fit well into a two-kingdom system of classification. The simple cells of prokaryotes set them apart in their own kingdom, Monera. Organisms in the other four kingdoms all have eukaryotic cells. Fungi are not photosynthetic, so they are given their own kingdom. The kingdom Protista is an assortment of eukaryotes that don't fit into the other kingdoms, mostly unicellular organisms such as paramecium, unicellular algae, and multicellular algae. The five-kingdom scheme divides living things into two fundamentally different groups, prokaryotes and eukaryotes. New data suggests that two kinds of prokaryotes diverged early in the history of life, and thus there are actually three major categories of organisms. A new classification system assigns more significance to the ancient split by creating a level of classification larger than a kingdom, a new category called a domain. The two domains of prokaryotes are bacteria and archaea. The domain Eukarya encompasses all of the kingdoms of eukaryotes. Taxonomists are in the process of sorting out various kingdoms of prokaryotes within the domains bacteria and archaea, and also splitting the protists into several kingdoms within the domain Eukarya.
All right, so hopefully that video kind of made things a little bit more clear for you. So a little bit more on phylogenetic trees. So the evolutionary history of a group of organisms can be represented in a branching phylogenetic tree. What this represents, a phylogenetic tree, this represents kind of a hypothesis about what we currently think is how all these different things evolved, where they split off, like who had for common ancestors, what traits their ancestors had, etc. So there's a few things we can learn from here. Each of these branching points represents the divergence of a species. So this is a speciation event. There was some sort of block to gene flow here, whether allopatric or sympatric. And at that point, we split into two populations, which eventually evolved into creatures that were like the otter and the badger, or creatures that were more like dogs. And then these dog-like ones, eventually there was a split here, and the wolves, this group here, the canis, split into what we call wolves or coyotes. There's a speciation point here. Now this obviously happened after this, but you can't necessarily say from a phylogenetic tree that exactly how long ago this was or this was. unless there's some specific extra stuff on here, we can't infer time on this, but we can say this first, that second within that that tree. So a little bit more about what we can see from a taxonomic tree. So here's all the different, or phylogenetic tree, sorry. Here's the different taxa or groups or species, whatever we're getting down to over here. So B and C here have a common ancestor at 4. So at that point, there was some common ancestor that they shared that has all the characteristics, and there was a split. Okay. If we go a little bit further back, we can find a common ancestor between A, B, and C. Okay. So these 3 right here are more closely related to each other than they are to these ones, because these ones share a more recent common ancestor right there. Okay. To find the connection between these guys and DEF, we got to go all the way back to here to find that common ancestor. So that's even further back. So they are related. Okay. At that point, we can come over here and find these 3. One quick note here is, you see how there's 3 coming out of 0.5? So there was a speciation event somewhere in here. When you see 3 together, the general consensus is that it's probably not a triple speciation event happening at exactly the same time, that we had 3 different populations that each separated and developed separately. What it's likely happened is that, there was one branching where, say, D and E came off, and then slightly later there was a branching between E that created F. But it's too close, we don't know. So it's a little bit of uncertainty there when you see that. And if you go back, all of these have a common ancestor way back here at 1. Okay. So what we can and cannot learn from phylogenetic trees, because they are limited, as is any scientific model, they show patterns of descent, not necessarily phenotypic similarity. So what's the difference there? Yeah, well, one, phenotypic similarity is, you have similar traits. While the, ancestors that, or organisms that had a common ancestor are likely going to share some phenotypic traits, they may not. If they got separated into very different environments, or for a long period of time, or, a very small population, like a bottleneck or something like that, they could have lost some of the traits that are similar, like birds and reptiles. Birds likely evolved from reptiles, but in Linnaeus's taxonomy, we have class AVs for birds and class Reptilia for reptiles, where if we were organizing it via a phylogenetic tree, it would be reptiles, and then birds would be a subgroup within the reptiles. And we don't have that. And there's some debate out there amongst taxonomists and phylogenists about which is a better way to do it. Some folks say we should do all phylogenetic. Others want to stick with what we've got. So there's some benefits to both. It should not be assumed that a taxon evolved from the taxon next to it. So here we shouldn't assume that B evolved from C. What we can say is that B and C have a common ancestor in the past, but we don't know, we can't say that one evolved from the other. We just can say they both evolved from that common ancestor. Phylogeny provides important information about similar characteristics in closely related species. That we can do. That's what it's useful for. Okay, so at this point, guys, I would like you to go to page 551 in your book and do the concept check 26.1, numbers 2 and 3. And then on Classroom, I do have a POGL activity for you called Phylogenetic Trees, Model 1. And if you're not doing this with me, you'll, in which case you'll do it in class, do it on your own. And if you have questions, come and ask me. And then we'll, on the other side, we'll talk a little bit more about phylogenetic trees. Thanks, guys. See you in a bit. All right, guys, I hope that practice went well. And if you have questions, talk to me and I'll help you out with it. The next topic for us is, how do we make phylogenies? How do scientists do that? In order to do that, phylogenists or systematicists or taxonomists gather information about the morphologies, the structures of organisms, their genes, and or their biochemistry. Sometimes they only have fossils to work with, sometimes they've got genes, sometimes they don't, sometimes they got biochemistry, so they just take what they can get to try to get as close as possible. Morphological and molecular homologies. Phenotypic and genetic similarities due to shared ancestries are called homology. So if there's similar structures and or similar genetics because they share a common ancestor, we call it homology, like homozygous, similar, right? Organisms with similar morphologies or DNA sequences are likely to be more closely related than organisms with different structures or sequences. So obviously, if you got more structures or genetic sequences that line up, you're more likely related than you are to somebody who's got fewer sequences that line up. I think that's pretty self-explanatory. Which is more accurate and reliable, the morphology or the molecular data? Likely the molecular data. It is the base code. Morphology can be misleading at times. Things can look very similar but not be the same. So that's why a lot of stuff has changed over recent history as we've gotten better and better and collect more and more genetic data. There's a lot of a rearrangement on those phylogenetic trees and in those taxonomic orders and taxons as we just get more information, which is what good science is all about. Okay? You do your best you can with what you've got. As you get new information, you can refine things. One thing that is a little tricky that we have to keep in mind is we have to sort homology from analogy. We have talked about homologous structures and analogous structures before in Bio 1 and a little bit earlier in this unit. So homology is a similarity due to shared ancestry. So this means this is divergent evolution, what we talk about as normal evolution. So you had a common ancestor, you had a speciation event, and now over time you've gotten a little bit more and a little bit more different until you're different species. The characteristics you share and the genes you share are homologous. Okay, they came from that common ancestor and then changed slightly over time. Analogy or an analogous structure is a similarity due to convergent evolution. So that's that dolphin and the shark we talked about before, right? They have different ancestors, like the shark's ancestor was a swimmer, but the dolphin's ancestor lived on land. But because they both eventually occupied the same niche in their environment, they had similar selective pressures, which over time we would expect would cause them to have similar adaptations. Hence the coloration, the body shape, the different structures that they have are analogous. They don't come from a common ancestor, so they don't denote relatedness. So just a reminder here, again, convergent evolution occurs when similar environmental pressures and natural selection produce similar or analogous adaptations. An ant leg and a horse leg they're both legs, but they're nothing alike. The way they work, their structures, everything else, there's nothing really similar about them as far as how they evolved. Same with a fish fin and a dolphin flipper. They serve a similar function because they're in similar environments, but they didn't come from a common ancestor with a fin. And then flight is a prime example. Flight is a really advantageous adaptation to any terrestrial environment. So it stands to reason that if it evolves, it'll stick around. So it's evolved many times in many different groups over time. And you can see these two wing types are very different in how they work and how they're structured. Superficially, they're big, they're broad, you move them, you fly. But how they do that, very different. Same thing with these two moles. They look very similar. Long body, big front claws, tough nose, small eyes, but they're really not related to each other very much at all. Other than the fact that they're mammals. Okay? They separated a long time ago. Bird and bat wings are homologous as forelimbs, but analogous as functional wings. So they're homologous as forelimbs, but analogous as functional wings. Can you explain that to me? Yeah, so if you go far enough back, it's like 350 million years, you can find a four-legged animal that eventually, became, split up a whole bunch of times, and one of those branches became birds, another branch became bats, way back when, okay? But then, from one of those descendants of that common ancestor, one of them, which was a reptile-like critter, a dinosaur-like critter, developed flying and used feathers to do it and a little bit different muscle groups to do it here. And then completely separately and millions of years later, a different descendant of that common ancestor that was still walking on four legs developed flight as well, but this time it did it with a stretched membrane. You can see how the finger bones come out extended to provide that surface area where the finger bones connected here, complete opposite, different things. So their limbs, we can see they've got scapula, collarbone, then they got the humerus, radius, and ulna, and the phalanges here. Same thing down here. So there's homologies as a forelimb, but the wing aspects of it, the wing functionality, came later and from in completely different ways. So the wings are not related. So the limbs are homologous, but the wing functionality is analogous. So you can see how we can evaluate morphological homologies. How do we evaluate molecular homologies? We use computers. It's really easy to use computers when analyzing this. So for instance, if here's the two different organisms, they're, what their common ancestors' original DNA look like. Now they're split, so they got different populations. And in this one, there's a deletion here. In this one, there was an insertion mutation there at some point in their past. Now this is what those gene segments look like, right? And on a surface inspection, they look very different, right? Nothing matches up here anymore. Down here, first part matches, the rest doesn't seem to match. But then you realize, oh, we can account for those mutations that happened, right? So here we can see that we entered in that GTA and that CA got shifted over. And here we can see that we deleted that G and these ones got shifted over. So we can see there's actually quite a lot of alignment here. And we can track where the mutations and the differences happen. And that allows us to see how similar or different these things are and where those changes happened. Okay. At this point, go and do concept check 26.2, number 1 on page 553, and go back to that POGO one, but this time do model 2, and I'll meet you back on the other side. So once we figured out what structures and traits and genes are homologous or analogous, we can use them to infer that phylogeny. And we can also take a look at traits that were either gained or lost during evolution to help us construct those phylogenetic trees and cladograms and figure out where these divergences happened. First, I want to mention cladistics. Cladistics groups organisms by common descent. So you're a clad if you all share a common ancestor that you descended from, right? So all these birds would be a clad because they all descended from a common, dinosaur-like thing that developed flight. So it's a group of species that includes all ancestral species and its descendants. And kind of like with taxons, they can be nested in larger clades, but not all groupings of organisms can qualify as clades. Sometimes we group them based on other things. Here, let me show you. So this is what we generally think of as a clade. Okay, so we've got these 3 modern day organisms, and it also includes, the common ancestors of B and C, and the common ancestors of all 3 of them right here. That's a clade. Okay, here's a group that includes something else, something over here. Here's a group that doesn't include all the ancestors of these. This is the one we're going to mainly be referring to. This is kind of like the equivalent to a taxon, except more based on evolution and ancestry as opposed to current similarities. Okay. Shared ancestral and shared derived characters. So within a clade, in comparison with its ancestor, an organism has both shared and different characteristics. A shared ancestral character is a character that originated in an ancestor of that taxon or that clade, depending on which system you're using. So a shared ancestral character is something that the common ancestor to all the other ones in that group had. Their ancestor had this, so they all share their ancestor's character. A shared derived character is an evolutionary novelty unique to a particular clade. So within this big clade that had, the ancestor back here, we have all these things here. Well, these two may have something new. Maybe they developed a third eye. This one didn't have 3 eyes. These ones didn't have 3 eyes, but these 2 developed a third eye. So that third eye would be a derived character. Okay. Let's take a look at a real example. So when inferring evolutionary relationships, it's useful to know in which clade a shared derived characteristic first appeared. So look at the turtle and leopard clade right here. So this little clade right here. What is a derived characteristic and what's an ancestral characteristic of that little clade? So within this clade, hair is a derived characteristic for the leopard, right? It, is not shared by the turtle. An ancestral characteristic for this clade would be something that both their ancestors shared, which is an amnion. So amnion would be a shared or ancestral characteristic, as would all these other traits here, because these are all traits shared by that ancestor. This ancestor had 4 legs, it had a hinged jaw, and it had a vertebral column, which this ancestor had, and these guys have. So all of these would be ancestral characteristics. This would be a derived characteristic. An outgroup is a species or group of species that is closely related to the in-group, which the in-group is the particular group you're looking at and studying is the in-group. The outgroup is a species that's really close, but not in your group that you're studying. And we can use that outgroup to make comparisons, to see what diverged within our in-group and when. So in this case, the outgroup in this particular, like this big clade we're looking at here, this big clade, the outgroup would be the lancet. Okay, it's the least related of these guys, right? Because you got to go further back to find a common ancestor. And all of these here have a vertebral column. The lancet doesn't have a vertebral column. It's got a spinal cord, but no bone around it. So this is the outgroup and allows us to see that, oh yeah, what separated here and what changed is that vertebral column. Now, some phylogenetic trees can have branch links that mean something. Remember I said earlier that don't assume the branch length means anything. The branches are just representing the connections. Well, you can draw phylogenetic trees where the branch length means something, but you can't assume that. You have to see it. It has to be clearly stated. Like in this one, these branch links represent the number of genetic changes that have taken place in a particular DNA sequence. So you can see here's the common ancestor for this whole clade right back here. And this Drosophila, look how long that line is. It goes all the way out to here. The human and mouse line only goes this far, which means what we can infer is there have been fewer genetic changes between our common ancestor with the Drosophila, fewer genetic changes between that common ancestor and us, and more genetic changes between that common ancestor and the Drosophila, the fruit fly. Other trees, the branches can represent absolute time, like chronological time, not number of changes, but chronological time. Here we can see, you can see about when the human and the mouse separated, or when the chicken and the human and the mouse separated, right there, whoop, right there. When we separated from the fish, or when we separated from the lancet and we developed a backbone, or when arthropods separated from us, all the way back 542 million years ago. Okay. And just a reminder that phylogenetic trees are hypotheses, guys. They are based on all the information we have, they're our best guess on to who evolved from who. One thing we can do with them, another thing we can do is we can hypothesize about characteristics of organisms that aren't around today that we don't know for sure. For example, so bracketing. We know that birds and crocodiles share a common ancestor way back here with some different branches of dinosaurs. Okay. Let's see, what is it? Oh yeah, I got it on the next page. So we can see what are the ancestral characteristics of the dinosaur and the birds. If it's an ancestral characteristic of both of those, the other things that branched from that common ancestor likely shared similar ancestral characteristics. Obviously, they would have developed their own new derived characteristics, but they would have shared those ancestral ones. Okay. For example, birds and crocodiles share several features. They both have a 4-chambered heart, which is good because that's a very efficient heart. 3-chambered and 2-chambered hearts are not very efficient. So it also makes sense that big things like dinosaurs would need a four-chambered, efficient heart. They both use song for communication. It's your opinion on who's got a nicer song. They both build nests, and they both brood or take care of their young in the nest. Those characteristics then likely evolved in their common ancestor, and that happens to be the same common ancestor it has with dinosaurs. Has the same common ancestor. So it's likely, we can infer from that dinosaurs brooded on nests and built nests. And it's also likely they had a four-chambered heart. And likely they communicated with song. Now, we don't have evidence for all that. Hearts don't fossilize very often. We can't tell if they sang, but we have found fossil evidence that they build nests and that they sit on those nests. So that's a little bit of evidence that this bracketing technique is useful with phylogenies. So here's a little video kind of going over that. I'll meet you on the other side.
By studying various fossil records, geologists have put together a geologic record. Evidence in the fossil record of large-scale extinctions and/or enormous radiation of many diverse new life forms defines the main boundaries within the geologic record. As you move through the geologic record, the names and dates will change. Also, a diagram at the upper left will show what is known of the history of continental drift, based on evidence dating from the Mesozoic era. During the Precambrian time, which began at least 4,600 million years ago and ended 542 million years ago, life began on Earth. The first prokaryotic cells and cyanobacteria appeared, as well as the first animals. The beginning of the Paleozoic era, 542 million years ago, marked the origin of most modern animal phyla. The first plants, jawless fish, bony fish, and amphibians evolved. Amphibians became the dominant vertebrate life form, while the first reptiles and seed plants appeared. By the end of the era, 251 million years ago, many marine and terrestrial organisms had become extinct. The Mesozoic era stretched from 251 million to 65 million years ago. At the beginning of this era, the reptiles radiated out as dinosaurs to dominate the landscape, along with the cone-bearing plants, or gymnosperms. At the same time, the first birds and mammals originated. As the climate and Earth's surface changed, flowering plants appeared and many organisms, including most dinosaurs, became extinct.
The Cenozoic era followed the Mesozoic, 65 million years ago, and continues through modern times. The beginning of the era saw the radiation of the mammals, birds, and pollinating insects such as bees and butterflies. The angiosperms evolved to dominate the landscape as most modern mammalian orders appeared. The primate groups evolved, leading to the origin of ape-like human ancestors, and finally humans.
All right, at this point, guys, I'd like you to do concept check 26.3, number 1, on page 559. There are no topic questions for 7.9, so don't worry about that. And we got just one more quick little thing. I'll catch you on the other side of doing that. Okay, so now the last little bit I want to do in this section of the notes, guys, is on topic 7.7, common ancestry. And I think we've covered pretty much everything we need to cover here for this topic in this unit and in past units. It's more of an exercise in kind of dredging up those memories and applying them to the concept of common ancestry. So let's take a look here. So evidence indicates the common ancestry of all prokaryotes and eukaryotes. So that there's some evidence that all prokaryotes and eukaryotes developed from a common ancestor in the very beginning of life on this planet. Can you think of some things that support that above statement? Can you think of some examples we've seen in this class so far that support the fact that eukaryotes and prokaryotes share a common ancestor? They have some common ancestral characteristics. All right, if you're not done thinking, pause it, but here are a few. We all use nucleic acids as our genetic material. Okay, so we have that common molecule, that common language of genetics there. We all carry out protein synthesis using ribosomes. Now, are there some differences in the details of that? Yes, but the basics are there. We use ribosomes. They read mRNA. They match up codons on tRNA and attach those amino acids, and we use the same amino acids. That's some pretty hefty evidence that we've got a common ancestor. There's also similar metabolic pathways in each, in certain ways that we use glucose for energy and convert it into ATP and glycolysis and things. So there's a lot of similarities there. There are more, but those are just a few that we've talked about so far, which is kind of interesting if you think about it. So we can make these connections with this common ancestry. There's also structural evidence then as well that indicates the common ancestry of all eukaryotes. So what are some derived characteristics of eukaryotes that derived after we split from prokaryotes? Or the other way you can think of it, what are the ancestral characteristics that all eukaryotes share? You can think of it either way. Think of a few of those for me, guys. Pause it if you need to. All right, what are some of the things you thought about? Some of the things I thought about was that we all have membrane-bound organelles, right? And those organelles have the same function. Mitochondria, nucleus, ER, Golgi bodies, all that kind of stuff, vacuoles, right? We all have linear chromosomes as opposed to the circular plasmids that you find in prokaryotes. We don't have those. We have linear chromosomes. And our DNA is broken up into chromosomes, not just one long strand. Similar cell metabolic pathways. So if you follow some of the molecular pathways, the GTP, the signaling stuff, a lot of that stuff is similar in eukaryotes. Our genes contain interons. Prokaryotic ones didn't. We've got interons. They don't. There's another one. And there are more, but those are just a few that I thought of. Okay. You might have some others. At this point, guys, do the topic questions for 7.7 on AP Classroom, and you'll see you for the next section of notes, part 5.